every cell in your body has the same exact DNA code, but yet we have stem cells, we have bone cells, we have fat cells. So how does a cell know what to do, what genes to turn on and what genes to turn off to actually become the cell type that needs to be to function? When we think about a lot of different diseases, say heart disease or other complex diseases, you have some genes that maybe should be set at 5, it's turned up to 11, and that turning up to 11 is what dials in that disease state.
All right, we're live on another episode of Life Sciences 360, and my guest today is Brian Cosgrove. He is the principal scientist at Tune Therapeutics. And Tune Therapeutics, if you haven't heard about them, they are orchestrating the power of Epigenome to treat some complex diseases. So we're going to talk about everything what's going on with Brian's role and Tune today. So welcome to the show, Brian.
Great, thanks for having me. Yeah, it's great to be here.
Yeah, so I saw that you were you were doing some research at Duke, and then you decided to join Tune Therapeutics. So can you walk me through that transition or what was going on there?
Yeah, no, it was a really interesting time. I think so. I had done my postdoc in Charlie Gershbach's lab at Duke University in the bioengineering department there. And when we were in Charlie's lab, there was a lot emerging at the time about new sets of tools using CRISPR-Cas technology, but not using CRISPR-Cas technology to cut, but using CRISPR-Cas technology as basically a way to go and put a protein at any location precisely in the genome, not cut, and bring along some protein payload to do something with. And so what we had been working on is tools to change the epigenome. And that really kind of spawned out a lot of where Tune Therapeutics started from, where these technologies that we developed in Charlie Gershbach's lab.
Yeah, and I'm not familiar much with, the approach to medicine and developing like epitherapeutic treatment. So if you were to simplify maybe a little bit more about epigenome and what exactly is epigenetic tuning, and I guess is that epigenetic tuning, is that why the company is called Tune? I'm just curious.
Yeah, no, exactly right of why the company's called Tune. And yeah, it's a great so the way that I like to talk about it is that, every cell in your body has the same exact DNA code, right? Every cell has the same written instructions, but yet we have stem cells, we have bone cells, we have fat cells. So how does a cell know what to do, what genes to turn on and what genes to turn off to actually become the cell type that needs to be to function? And it's not through changes in the DNA, but through changes in the epigenome. And so essentially the actual piece of DNA is wrapped around a protein called a histone. And that protein kind of has different ways to regulate the DNA. And the way I like to think about it in the most simplistic way is it could kind of pack down the DNA so tight that nothing can get into it. So it's like throwing the clothes in the corner of the closet that you never go to. At some point, you're not going to need them. They're back there. And the things that you're going to need a lot to be a fat cell, it's keeping them open, letting proteins come to turn on those genes and basically saying, hey, I'm a fat cell. I'm never going to need any of these genes over here for, making minerals for bone. So I'm just going to turn them off. And so there's a set of just different chemical marks that could get put on the proteins around DNA or on DNA itself. And that just kind of helps each cell turn on or off different genes. And so this idea is, can we actually go and regulate and reprogram those different marks around the DNA without necessarily touching the DNA sequence itself and start to actually change gene expression in a way? And your point about tuned is precisely right. It's about, tuning expression. And I think a lot of the times there's a lot of different focus on monogenic diseases, right? And especially at the first wave of CRISPR-Cas therapies, it's what monogenic diseases can we go and make one cut, fix a broken piece of DNA. But when we think about a lot of different diseases, say heart disease or other, complex diseases, it's mostly the case where you have some genes that maybe should be set at 5, and it's turned up to 11. And that turning up to 11 is kind of, what dials in that disease state. And we don't want to turn it all the way off or all the way on. We kind of want that precise bit of tuned expression that is the healthy level. And so it's about thinking about how can we actually use these tools to start to tackle a more complicated set of questions in biology and complicated set of diseases that are more common that everybody is kind of exposed to.
Yeah, it always fascinates me because I work within consulting, but I mostly work within quality, compliance, and technology projects. I do get to work with a lot of scientists because technology is being used in all different areas of the industry. But till this day, every job I've ever had in life sciences with every company just fascinates me how complex the human body is and how many different alternative pathways there are for scientific research to bring medications. Every time I talk to a new CEO or a new company on this podcast, it just blows my mind like how they're going down this rabbit hole on one point and figuring out, like, for you explain about epigenetics. So it just fascinates me that the human body is looks simple, but like within our different cells and different there's so much machinery inside the body. It's just fascinating.
It's definitely every day you learn something new in this research where it just kind of blows your mind that this whole pathway even existed and how did this even come to be.
Yep. So let's dive into one of your programs, HPV, because I know, from what I understand, there isn't cure for HPV right now. So, can you tell us a little bit about HPV in general, and what are the current options, for patients with chronic HPV?
Yeah, and maybe even as just a bridge before that, I just got done talking about all these common and complex diseases, and it's kind of interesting to pivot and be like, okay, let's talk about a viral infection,
Yeah.
and really, it's kind of an interesting story therein. But I'll talk a little bit about HPV to start with, and maybe talk about some of the, to your point earlier, about the cool biology that
Yep.
kind of
Yep.
lies within. But the idea is that I think many people are exposed in their different vaccination schedules to hepatitis B. That might be where the main familiarity is, but it is an infection that affects hepatocytes in the liver. And while most individuals can clear this infection normally, in a subset of these individuals, this becomes a chronic health condition of the liver. And what happens is essentially you have these infected hepatocytes that become these viral production factories, which ends up damaging the liver, leading to liver damage or failure or hepatocellular carcinoma or cancer. And while we do have really good vaccines and therapeutic vaccines, and I think really over the past 20, 30 years, we've seen really great vaccination campaigns, there's still currently, I think, around 350 million people worldwide that have chronic hepatitis B. And so there's this really interesting subsection where you say, yeah, there is a vaccine for this virus, but we have a huge patient population that has this unaddressed clinical need where they have this latent viral infection. And even if they go on antiviral medication like nucleoside analogs that are commonly used to kind of prevent viral replication, there's still this very slow liver disease progression that, patients usually get this disease a little bit younger in life, and there's this burden of progression of liver failure, and whether it's cirrhosis or cancer, you really have quality of life issues that pop up downstream with this latent viral infection. So there's a really huge unmet need globally in this population of what do we do about these 350 million people that have
Mm.
chronic hepatitis B, and that number, hopefully we could get down with other strategies, about therapeutic vaccines, in other words, but it's always hard to deploy those all across the world. So the question is, we have really a finite duration therapy for anybody that can be both prophylactic, but also, be able to treat people with chronic hepatitis B.
Mm.
And
Mm.
and yeah, and maybe I'll pause a little bit there, but I could also go into the biology of it as well. But it's a really, really interesting case for epigenetics.
Yeah, it is. And so yeah, I want to hear more about the biology side of things as well. But so it's interesting that I also wanted to ask you another question, which is, when you came into Tune from your previous work in the research space, did you always wanted to be in the life sciences industry, or were you like thinking maybe I should be in academia and see, maybe be a professor or do some other stuff, or how did you end up coming to Tune?
Yeah, no, so You hit the nail on the head there, where I have this work that we've been doing in Charlie Gershbach's lab, and I think right around the time this was when COVID hit in 2020. There's a
Yeah.
lot of time at home, a lot of time with family, and you start to think about, for the whole time I was like, I really head first into academia. Can't wait to do academic research. That's where real cutting edge stuff is happening in the field, and that's what I want to do. And I think slowly over time that kind of evolved to think about, is it the one problem that I was working on in the lab that I really cared about and wanted to work on this problem for the rest of my life? Or were there ways that I could actually take this technology and actually do something with it therapeutically? Is it that I don't care
Mm.
necessarily what the technical problem is? I could learn about new diseases, new contexts, but I just want to do something with all this information that we had. And what we had in Charlie's lab was this really great tool set that we could apply to almost any gene in any location in the genome and really start to turn on and off gene expression. And at that point, the question becomes, what can we actually do with this therapeutically speaking? There's a lot of diseases that could probably benefit from really precise programming here. And so I think that was this moment where it was academia or industry, what to do. And, a lot of time at home with family trying to figure out what was the right path. As we mentioned, I'm in the research triangle area, and trying to think about the academic cycle of being like, oh, you can move to anywhere else in the country, we'll see where you get an offer, who knows where you go, uproot your entire life, family and kids, move somewhere else, and take this job. And there was a lot of interest in a lot of the research happening in the research triangle at the time, and there's a lot of VC activity. And shortly as I was making that decision, a few researchers in Charlie's lab spun out this company, Tune Therapeutics. And there was a bunch of lunches where it's like, Brian, we could do something with this technology. We could be really programming disease with this. Is this something you'd be interested in? And I made the hop pretty early on in the life of Tune to come over and start to work on actually kind of programming different therapeutics with this type of technology.
Yeah, it's interesting because I haven't really talked to a lot of people who come into a company at an early stage. So, how many employees are at Tune right now?
So
Yeah.
I think we're at about 100 employees right now. So about
Okay.
50 at our Seattle site, about 50 at our Durham site. And when I joined, I think we had about 20. So it was pretty early on in the life cycle.
Right. So, if you compare to when you joined at 20 and where you are at 100, because that's such an amazing, like the first 50 or the 100 people. I haven't joined a company that early. So that's why I'm asking you this question. Like, what is it, if you were to, phrase it in a few sentences, like from where you joined in the early days to where it is now? You want to comment on that?
Yeah, I think, it's kind of funny. It's in some ways it's a lot of different, in some ways it's not different at all. The parts that's
Mm.
not different at all is it's still that we have that discovery feeling that kind of like permeates the entire company. And I think this idea of at 20 people is, hey, let's go and find what diseases make the most sense for this tool set. Where can we make the biggest impact right away? And we kind of had this really good energy in the company at that time of like, hey, this is working in this area, this is working in that area, this is working here. And when you had people come on from industry, it was very funny to hear their takes of saying like, this doesn't happen all the time. You can't apply these tools to all these diseases and see that there's generally success in a lot of different areas. Usually you just have that one clear path where it works, and it's kind of a beam of light shining down, and that's the way you go. So the early days, it was trying to figure out how to get these tools to work, where they work the best, and really kind of playing around with that. And I'd say the biggest shift now is just thinking about how could we deploy these tools as quickly as possible and get them to the clinic. And that's really been the biggest kind of shift in thinking that we've all gone through, is how do we keep that pace up from that early discovery and keep that excitement and passion, and really keep getting everything kind of working in tandem, whether it's delivery tools, whether it's the protein technology itself, and getting all these different people in the same room with different sets of expertise, and getting them on the same page and having everybody go towards that common direction. So it's been, I'd say the biggest shift is just how to quickly pull people into the loop, how to get people focused and on the right kind of path. But the part that's the same is that energy and excitement and that push of that feeling of this could really be a therapeutic, and the excitement of being able to help patients that have this disease, I think is still there. And that's the fun part. And that's what keeps it exciting every day.
Yeah, it's I've had some friends who are in similar position as you know, who've like joined a company in the early days, whether it's being in reg affairs or helping with submissions and stuff, or being in the science or manufacturing side of things. Yeah, and I've seen more. I worked in more companies that grow from, like, let's say 200 to 500 and 1,000 ish employees. So I've seen some, you know, differences in that frame, but I haven't seen the, you know, before 100 frame. So that's still very unique that, you know, you as you mentioned, you were having lunch with colleagues at Duke, and you started, you know, got into this company, and here you are with 100 people. So that's pretty interesting.
Yeah,
Uh,
it's
yeah, I wanted to talk about the Tune 401 product. But before I go there, can you explain to the listeners what the Tempo platform is? Because I was reading a bit about it, but couldn't really get a grasp of what it really is. So do you want to shed some light on that?
Yeah. So the Tempo platform is kind of the evolution of this tool set that I talked about at the beginning of the podcast in Charlie Gershbach's lab. But it's this idea of how could we program a protein to go to a very precise site in the genome, sit just there, not in any off targets or anywhere else. Don't cut the DNA, just sit there and bring along a protein payload and be able to go and remodel the epigenetics of that given site. And so in this case, we've developed a lot of tools that actually could take a site that's on and open and transcribing a gene, and it remodels that protein DNA structure called chromatin to kind of compact it down and turn it off and put it aside. And what's really exciting about the Tempo platform is it's a lot of different epigenetic domains, a lot of different DNA binding domains, and it's both a therapeutic, and it's also the idea that these platforms could actually be used to do a lot of high throughput screening. And so the idea is that you could put in libraries of target sites and very quickly figure out where each of these modifications is the most efficacious towards changing gene expression. And so really, it's both the downstream tools that we're using in the clinic, but also I think part of what's enabled us to move so quickly is it's very similar tools that we did all of the early discovery with. And we set the parameters to say, okay, let's use these same systems from start to finish. And I think that's something that's lost in a lot of companies, that you move from system A to system B to system C. And every time you move to one of these new systems, there's that integration kind of issues that pop up. And so the Tempo platform really enables us to go from this high throughput discovery all the way down to precise therapeutic epigenome modification with very similar tool sets.
The reason I asked you that first is because I wanted to just get a good understanding of that before we go and talk about Tune 401. So now, if you want
Yeah.
to maybe expand a little bit about how Tune 401 works using the Tempo platform for the HPV treatment. So can you explain a little
Yeah.
bit to, yeah.
Yeah, it's just pretty interesting because, the story of how we started to work on hepatitis B was kind of interesting, and it bridges that early discovery into, a therapeutic, as I just mentioned. And we had started off and really looked into potential targets where it seemed like, epigenetics were a driver. And what was really, really interesting was, you know, as I dug into the biology of hepatitis B, it's this really interesting epigenetically regulated virus. And so what happens is when a hepatocyte gets infected by the virus, there's almost this mini chromosome that forms. That's a circular double-stranded chromosome, not completely just like a plasmid, that forms, and it lets it sit there latent in hepatocytes. And what's interesting is that double-stranded circular piece of DNA actually becomes this like Franken chromatin. So basically,
Hmm.
it's part viral, part human. So it takes some of the human proteins and uses those to regulate its transcription. So it kind of wraps its DNA around those histone proteins. And some of the viral proteins also kind of act like pseudo human proteins and helps it regulate the transcription from the virus. And so it's really interesting as you dive in that the hepatitis B virus is completely regulated by changes in epigenetics, just like host human genes. But it's also a little different because all of these viral proteins kind of have their own rules. And so it was really interesting coming from the field of studying host genes. And all of a sudden you start to look at this hepatitis B virus and be like, well, the same general rules apply, but it also has its own unique twist of what is, possible. And there's a unique set of epigenetic rules that govern the virus. And so early on, we did a lot of different screening and found different regions in the virus that we could actually go and precisely target with our epi silencer, we're calling it, but basically an epigenome editor tool that comes in to close down that DNA, pack it down, and turn it off.
Hmm.
And what we found was that there were some master epigenetic regulators in this viral life cycle. And what we could do is actually go in and turn those off. And that would actually cause that virus to basically sit there completely inactivated, just like some of the other host genes and cells that we talked about that were turned off by epigenetic means. And digging into it, what was even more interesting is in more recent work, there's a small subset of patients that there's evidence that they actually silence the virus and they're stable off treatment by their bodies kind of spuriously over years and years and years and years, putting on different epigenetic marks on this virus to try to turn it off. And so
Mm.
it's this kind of battle over all these years between the body that's natural defense is to try to come in and put some epigenetic marks on this DNA to turn it off, and the virus that's programmed and evolved itself to remove any of those epigenetic marks, that the body's trying to use to turn it off. And so with our tools, what we could come in and do is precisely go into those DNA sources and say, hey, we're going to really completely program that aspect of biology, put on these epigenetic marks, and then just shut down the virus. And that's what really could lead to a functional cure for this disease is that epigenetic programming and the durability of epigenetic state and keeping the genes off.
Mm. So you were mentioning earlier when you said that this technology can be used for curing different other multiple other types of diseases. So outside of HPV, are there any other areas where Tune is trying to maybe do the early testing and see if there's any promise?
Yeah, I think in terms of early testing, kind of mentioning that early development cycle, we shared some data about using these tools to silence PCSK9 in the liver. And PCSK9 is a regulator of cholesterol,
Mm.
and it's been a target for monoclonal antibodies and other gene therapy approaches to basically silence this PCSK9 gene. And there's been some genetic studies that show that patients that have more control over their cholesterol have less PCSK9 expression. And what we had done is actually found that the similar idea of going in and programming in this repressive epigenetic state worked really well for PCSK9. And it's actually kind of intertwined really nicely with the HBV story in that we have monkey data and NHPs
Hmm.
that shows that we could have a single dose of these epi editors that has shown durable repression of PCSK9 now out past a year from just a single dose of these epi editors. And that NHP data has actually been kind of tied to our Tune 401 HBV epi silencer because there is no large animal model for hepatitis B. It can only infect humans and chimpanzees. And so you have to rely on these proxy models to say, how is your dosing? And can we find a gene that's regulated very similarly? So we've shown a lot of work for how these tools could be used in the context of cardiovascular disease, in liver targets for long-term repression of PCSK9. And we've also shared data for ex vivo therapies where there's a lot of different options in terms of CAR T therapy or iPSC derived work where we're able to kind of layer onto these technologies and add small changes in genes that make them more effective. So it's really a unique problem set where we have a mixture of ex vivo and in vivo, and there's a lot of value add to each of these places, and here delivery tools become a really interesting consideration.
Yeah, and as you mentioned, just the potential in various other areas and other types of therapeutics. It sounds like that's what makes this technology that you have at Tune more versatile because it's not just confined to one area. It can be applied to multiple different domains. So sounds like it's a promising future for the company and also much excitement for you to be working as part of the company there for Tune 401. So where, like, I know just doing research that the company had shared some data in end of 2023 at a conference. But when does the company expect to start any human clinical trials for Tune 401?
Yep. So right now we're shooting towards dosing the first patient by the end of 2024 and seeing some clinical readouts in 2025. And so we've really, as I mentioned before, part of the flexibility of this Tempo platform has been really moving from some of these initial discovery workflows to the clinic in a really, really rapid pace. And that's been enabled by both these epi silencers and this technology, but also just in advances in delivery technology and you know, the fact that LMPs that we're using for Tune 401 as the delivery vehicle have had such great clinical success so far in terms of being able to really target precisely different cells in the liver in a non-viral approach. And it's been really exciting to see kind of the epi silencer part of Tune 401 merge with the LMP part and enable really fast translation to the clinic.
And if you based on, I'm sure you are having these discussions internally with your colleagues, because dosing the first patient is a huge milestone for any company, and I'm sure a lot of people at Tune are working towards that. In any of your discussions with your colleagues, do you foresee any challenges that are coming up, and what is your team doing to overcome them to make sure that you still meet the end of 2024 timeline?
Yeah, I think obviously manufacturing is always the biggest kind of lead time, and trying to make sure that you could make a product that you've characterized really well is going to be safe and effective. And so that's where we're at now, is working towards some of those milestones
Mm.
to be able to dose the first patient ex-US. And I think really what we've also been focused on is, once you start getting those readouts from those first patients, hepatitis B is a really complicated disease. And so, trying to have the right biomarkers and trying to understand which of the different viral depots you're hitting and what is coming out in that patient, it's kind of relying on being able to have multiple shots on goal and being able to detect the readouts that you're looking for. So it's kind of a mixture of really good planning on the clinical end and having strong clinical collaborators, but then also really just trying to go through and understand, what it is like and what is the variability in manufacturing a product at scale.
Hmm. And for manufacturing, just out of curiosity, is it going to be in-house, or is it going to be partnering with virtual, or a mix of both? Do you have any
Yeah,
comments on that?
Yeah, so right now it's with partners, and I think part of the excitement is really relying on expertise that both internally and externally that could
Yep.
work together to get it to clinic quickly. And so I think a lot about this and a lot about the Tune story itself is, as I mentioned, we really started with this epi silencer technology, but it's how do we pull people in that have really great expertise and could really help us get this kind of base technology into the clinic as quickly as possible. And that's really been exciting. And we've shared that we've partnered with Acuitas for the LNP technology, and they have a lot of experience manufacturing LNPs for COVID. And they've been great partners to work with and really, getting this to the clinic.
Okay. And do you know of, as you're doing your work in this area and presenting at different conferences, again, this is just out of curiosity, do you know any other startups or companies who are also doing research in this space, sort of very similar to the area that you are playing in?
Yeah, there's a lot of different approaches that I think are out there. None completely like Tune
Okay.
in terms of their approach towards the technology, but just a few things to highlight that there's a lot of different epigenetic modifications, and so I think you'll see there's other companies in the space
Hmm.
just broadly of epigenetic non-DNA programming that are looking at different techniques and looking at different epigenetic modifications to treat disease, whether it's looping back to certain regions in 3D space in the chromatin. And I'm excited to see just in general how this space grows. I mean, we've seen that there's a lot of different gene editing companies. We've seen that there's a lot of different base editing companies. And I really think that what's exciting about epigenetic editing is there's more diseases out there that are, addressable by epigenetic editing. And I think Tune has really been at the front of pushing this forward, and I'm just excited to see how the field grows in the therapeutic space, because there's a lot of diseases where this technology is really powerful in being able to actually have an effect for a wide variety of diseases.
Yeah, yeah. So that, that again goes back to the point, like I was mentioning earlier, where you said that, this technology has a potential to, you know, find treatments for a wide variety of diseases. And, every company, like you're mentioning, it's promising to see that there's going to be other companies who will also make minor changes, fundamentally use the same epigenetic tuning, uh, you know, uh, model, but also make minor changes to come out with new therapies. So yeah, it sounds like an exciting space to keep an eye on, and I'm sure I will hear about other companies in the next few years as they go, from lab to patient. So
Yeah, and it's a space where I think it's been exciting that you could have uh, broad applicability even in terms of where you deploy these technologies to, and
mm,
that we've had a lot of success with this LNP mRNA nucleic acid delivery, and it's really something that I feel like is going to be able to be a therapy that we could make globally, that you could deploy globally, that doesn't rely on really complicated AAV to kind of scale and produce and deploy globally. And really excited to see how we take something that's really broadly applicable to a lot of different disease states, but also something that has been shown to scale in manufacturing, with the COVID vaccines and everything else, that you could deploy a nucleic acid therapy globally and have it actually be an accessible therapy. And I think when you think about a viral disease with a real global footprint, we're kind of excited about both that you could pair that epi-silencer technology with broadly deployable nucleic acid LNP therapies.
yeah, yeah, and it's, I can sense the excitement from all your responses and how well you explain this topic. So I know we're almost near the time to wrap this up, but before we leave, I wanted to ask you a couple of closing questions. Uh, you started in at Tune in 2021 at some point, I think mid or late 2021, and you've been there now, coming to the first milestone, end of 2024, when you actually get to take this into the first phase of clinical trials. What is the most rewarding part of all the work that you're doing at Tune?
I I think it's being able to take all of your scientific knowledge and all of this work, in academia, developing these tool
Mm,
sets, and actually being able to use them to make a difference. And I say that because one thing that I always keep in mind is my wife is an ICU nurse, and every day she comes home and it's this interesting. I've made a difference in somebody's life, and it's a
yep.
really nice pivot to go from I'm toying around with pipetting small volumes of clear liquids in the lab, and be able to say I'm actually taking that expertise, and maybe it's a longer game, but really hopefully using it to improve the lives of patients. And it definitely adds a sense of purpose that wasn't quite there before, and a sense of a driver pushing you to kind of go to the next steps.
Yeah, yeah. No, it reminds me of some of the conversations I've had with my wife, because she's a physician assistant and she used to work at, office settings and stuff like that. But when we moved to RTP area, she started working in clinical research. So now she's, a sub investigator. She does a lot of like patient enrollment. So now she's seeing this completely new side of the industry, working with, companies who are doing these trials, having meetings with them, understanding their strategy of how to run the trial, enroll the patients. So every time she tells me like, oh, this is such an interesting space, like I had no idea I could work in this as a PA. So yeah, it's always nice to have, somebody who's understands a little bit of what you're doing, because if, I guess, if you have a spouse that's in a completely different field, I guess that's also okay, because you need some change from the day to day that you hear. But yeah.
Yeah, no, I mean, I think it's really exciting to be able to, somebody takes all of it goes away from the patient so quickly. And, especially as a PA, having somebody that has that patient focused experience and brings it to their clinical trial design, because a lot of times patient compliance and issues like that are where trials kind of very
Yep.
quietly fail, right? You didn't have the right site management. You didn't enroll the right types of patients. You didn't, and really thinking patient focused. It's great that she could bring her expertise there and really kind of bring it to some of these drugs.
Yeah, yeah. I've joked with her because a few times, like she was like, "Oh, we are getting audited by so and so company. Like, what should we do?" And I I joked to her saying, "Maybe you should ask your manager to send me an email, and I'll send them a proposal, and then I can tell you what they can do." But yeah, it's
Starts with good quality early on in the life
yeah,
cycle, right? This
yes.
is where it starts.
All right. Thank you so much, Brian. This was an amazing discussion. I love everything, your excitement, your passion, everything you're doing. Any final words for the audience? Where can they learn more about Tune or just, keep in touch with you?
Yeah, you could go to our website and learn more about kind of the technology and how we're deploying it and how we really think that this is going to move the needle for hepatitis B. And I think we're just really excited about this finite duration therapy, a single therapy that could really move the needle to really help patients reach a functional cure. And just excited, as you mentioned before, about going from these viral technologies to seeing what other diseases, we could really show that epiediting plays a huge role in being able to control. And so really appreciate the chance to talk about this technology. Hopefully I did a good enough job explaining
Yep.
epigenetics and why everybody should really be focused about not just turning genes all the way on or off, but, being able to tune it in the way that you want it to go.
Yeah. And I take that you're on LinkedIn. So if people want to connect with you or anything, you're active there.
Definitely. Yep. That'd be
Yeah.
great.
All right. Yeah. So once again, thanks. I know you're in RTP area, so maybe whenever time permits, we can grab a coffee or lunch. But
Yeah.
I'll be rooting for Tune. I'll be keeping up with all the updates, and who knows, maybe we end up working together somewhere down the line when you go into more into GXP phases of development. So
Yeah, that'd be great. And would love to catch up more sometime in person. So thanks for having the virtual meet and excited to maybe meet you in person at some point.
yes, thank you, Brian. Appreciate it.
Thanks, Harsh. Appreciate it.